Hello, good morning, everyone. Thank you for joining. We'll get started in just a moment and let folks hop on in. Good morning. I see folks trickling in. Hello, good morning. All right, let's get started. So thank you so much for being here today. We're here to hear from our researchers on ocean acidification and hypoxia research that's been going on. My name is Tanya Torres, and I'm the program coordinator with California Sea Grant, based in San Diego at the Scripps Institution of Oceanography. I'm joined here today by my colleague with the Ocean Protection Council, Kyla Kelly. I'll let her introduce herself.
Hi, everyone. Kyla Kelly, and I'm the Water Quality Program Manager for the Ocean Protection Council.
And again, we're here to share findings from four funded projects that have been working over the last couple of years to improve the state's understanding of ocean acidification and hypoxia in California's coastal oceans. Thank you so much for being here. We kindly ask that for the duration of the webinar, you just keep yourself muted, and if needed, camera off for bandwidth, but I think we're okay today. And we'll go ahead and get started. So today, our agenda, Kyla and I are going to give a quick background and context from both of our programs, California Sea Grant and Ocean Protection Council. And then after a quick intro, we'll go into our presentations. We have, like I said, four researchers here today to share four different projects. They'll have 15 minutes to present each, and then we'll have 10 minutes of Q&A after each presentation. And then following the four presentations and Q&A sessions, we will have another round of discussion or more time for Q&A before we close out today at noon. Um, all right. And so just a little background about California Sea Grant for those who are unaware. Our mission is to provide the information, tools, training, and relationships necessary to help California conserve and sustainably prosper from our coastal and marine environments. And we do this by providing grants and funding to the larger marine and coastal research community in California. But we also provide fellowships to train and mentor the next generation of coastal researchers and managers, and we prioritize working with and for communities through our extension programs and our communications and outreach. And so to learn more about our program and our other funding opportunities, you can visit our webpage or sign up for our newsletter. And Kyla, if you could be so kind to add that link into the chat, that would be great. Um, and then
Okay,
Oh,
just adding to the chat simultaneously. Okay. Um, Let me just hop around. Okay, so for those not familiar with OPC, the Ocean Protection Council, we are a cabinet level state policy body within the California Natural Resources Agency. The mission of OPC is to ensure that California maintains healthy, resilient, and productive ocean and coastal ecosystems for the benefit of current and future generations. The solicitation was intended to meet OPC's 2020 to 2025 strategic plan objectives, including 1.2, which is to minimize causes and impacts of ocean acidification and hypoxia, and objective 1.3, improving understanding of climate impacts on California's coast and ocean. It also advances the research priorities laid out in the California Ocean Acidification Action Plan. And finally, the solicitation was aligned with recommendations made by the California Ocean Acidification and Hypoxia Science Force report called Enhancing California's OAH Monitoring Network, which was a joint effort led by OPC and OST, the Ocean Science Trust. Next slide, please. Just a brief overview of OAH at OPC. OPC's OAH program works to protect ecosystems and coastal communities from changing ocean conditions by improving our understanding of impacts to California's ocean and coastal ecosystems, identifying opportunities for actions, and building sustained support to address this problem. So the research we funded with California Sea Grant for the solicitation supports these program goals. And I want to talk about some of the other work we're doing to support OPC's OAH program. First, our new strategic plan reinforces our commitment to addressing OAH as objective 1.2 aims to limit the causes and impacts of ocean acidification, hypoxia, and harmful algal blooms. So we will be working to implement that objective through the end of the decade. Last year in March, the Council adopted a resolution on ocean acidification and hypoxia to elevate and communicate the state's commitment to addressing this water quality concern. The actions within this resolution reflect and build upon work that's already ongoing at OPC to understand and mitigate OAH, given the potential for it to cause large or irreversible effects on California's coast and ocean. Lastly, we are funding research to support more state actions on OAH, and we're here today to hear about the to hear about the research that OPC and Sea Grant has previously funded. And we recently opened a solicitation for monitoring research and modeling projects to support ocean acidification, hypoxia, and marine harmful algal bloom management in California. So Tanya, if you could drop that in the chat, thank you. We're not going to talk about the solicitation today. You can find a lot of information about it on OPC's website, and we held a webinar yesterday, which a recording will be available for soon. But this new solicitation, as well as the 2022 solicitation that we're here to talk about today, is intended to build this knowledge to support state actions. Next slide, please. Okay, jumping back to the solicitation we're here to talk about today, I want to note that it was funded by Proposition 68 funds. This was approved by voters in June 2018, and the funding appropriated to OPC is intended to protect and restore the state's natural resources for the benefit and enjoyment of future generations, as well as support projects that assist coastal communities with adaptation to climate change, including those reliant on commercial fisheries. And climate adaptation projects include those that address ocean acidification. And with that, I'll hand it back to Tanya.
Thank you, Kyla. So like Kyla mentioned today, though, we are gonna hear from our 2022 researchers who were funded back in 2022 and have mostly completed their research projects. And so just a little recap on what those research priorities were. They were focusing on funding monitoring research and synthesis projects that will enhance the state's understanding of ocean acidification and hypoxia on biological vulnerability. And these projects were selected to ultimately provide the state resource management agencies and local jurisdictions with data necessary to protect marine biodiversity and water quality, advance coastal adaptation efforts, and support climate-ready fisheries. And so today's speakers, we've got Cheryl Logan from the California State University, Monterey Bay. I'm going to give a talk on their project titled Assessing the Potential for Rapid Adaptation to Climate Change in Rockfish. Then we'll hear from Eric Yorkstead. Apologies if I didn't say that right, from Humboldt State University, on their project titled Advancing OAH Science Off Northern California, a critical expansion of monitoring and research to quantify OAH exposure, assess ecosystem impacts, and support model development. Then we'll hear from Christina Frieder from SCORP, sharing their project titled Multi-Stressor Tools to Interpret Effects of Acidification, Hypoxia, and Warming on Southern California Current Marine Calcifiers. And then lastly but not least, we will hear from Alex Harper with the Central and Northern California Ocean Observing System, or CENCUS, at California State Polytech University, Humboldt, on their project titled A California OAH Portal to Enable Synthesis and Understanding of Statewide Status and Trends. All right, that was a mouthful, but we do want to just have a little disclaimer here that please note that some of the findings presented in this meeting may be preliminary and not yet peer-reviewed, but we want them to be shared to foster transparency. We just encourage caution interpreting the implications until research is complete and peer-reviewed. We will be having Q&A after each speaker, and we would love for you to add any questions to the Q&A function You might have to click the More button to find it, but also use the chat if necessary. And again, just thank you for joining. If you have any questions for Kyla and I, here's our emails. But with that, we can go ahead and get started with our first speaker, which would be Cheryl Logan with California State University, Monterey Bay.
All right. Thank you. Can you see my screen? Okay, excellent. Well, good morning, everyone. Thank you so much for coming to the webinar today. I'm really excited to share the results from our project on behalf of our entire team here. Our project, again, is entitled Assessing the Potential for Rapid Adaptation to Climate Change in Rockfish. And I'll go ahead and get started. So this is the outline of my talk presentation today. I'm going to spend just a short period of time talking about background information. We're all here for the OAH webinar, so we know a lot about the changes in OAH that are occurring in the California current. I'll explain why we're interested in these effects on rockfish, and then I'll jump right into the objectives that we had as part of this grant and the results. And these are preliminary results that are not yet published, so keep that in mind. Our first objective was to examine how OAH alters rockfish reproduction and larval health. Secondly, we tested whether maternal exposure to OAH conditions during pregnancy could potentially boost offspring performance as a mechanism for rapid adaptation. We then looked at linking real-world variation in OAH conditions to rockfish reproductive success in the wild, kind of trying to confirm our laboratory results. And then fourth, we integrated some of our laboratory results into a stock assessment model to better understand how these changes in the environment might impact populations and use this model to better inform management. So jumping right in, we all know that upwelling occurs in spring and summer months along the California coast. And during upwelling events, we see a decrease in pH and dissolved oxygen levels in the nearshore environment. With the background of ocean acidification and deoxygenation occurring under climate change, we know that these waters are likely to become even more relatively acidic and deoxygenated in the future. For nearshore organisms that are living in these environments, it's a double-edged sword because with upwelling, we get an increase in productivity and more food availability. However, it makes it more difficult for these organisms to breathe if there's less oxygen in water and adequately regulate the pH inside of their bodies, which is really important for physiological function. And we're particularly interested in the effects on rockfish because many of them inhabit that nearshore environment. They're an important group of species as a commercial and recreational fishery. They're also important for the ecosystem as prey items, for seabirds and top predators. And there's more than 65 species that live in the California current, and they all have slightly different life history traits, some of which some groups of rockfishes spawn during the spring period, which coincides with upwelling. So we're particularly interested in those spring spawning rockfish that might be more impacted by upwelling and climate change in the future. And lastly, we believe that this group of species might have low adaptive capacity in terms of their ability to genetically respond to these changes in climate because they are very long-lived and late to mature. And these traits make it such that they might have a harder time keeping pace with climate change. So we're particularly interested in any other mechanisms they might have in order to rapidly respond to changes in ocean chemistry. So I'm going to jump right into our first objective, which was to examine how OAH alters rockfish reproduction and larval health. So for this first objective, we performed a series of lab experiments with the focal species of gopher rockfish. Gopher rockfish are a spring spawning species. They're also highly abundant and easy to collect and hold in the laboratory. So we collected gopher rockfish off the coast here in Monterey Bay, brought them into the laboratory at Moss Landing Marine Labs, as shown in this picture up at the left, and held them under either ambient, moderate, high, or extreme OAH conditions that correspond to present or future day conditions that might be expected under upwelling. We held them during their entire gestation period. These are live-bearing fish. And then after they gave birth to their larvae, we also measured the potential effects on their larvae immediately after birth. So the response variables we looked at were metabolic rate, and blood chemistry and fecundity in the moms. And then we also looked at how these exposures might impact larval morphology. So these are some results from the first objective, first looking at metabolic rate. So what we found was that just looking at pregnant versus non-pregnant females, that the pregnant females had a higher resting metabolic rate, as shown here in the top. And this suggests that they have higher energetic demands during pregnancy, which just means they might be more susceptible to the effects of environmental stress. In addition, we found that under OAH conditions, on the bottom graph here, we're looking at maximum metabolic rate, and we found that OAH reduced maximum metabolic rate in these adult females. This could be problematic because it's going to make it harder for them to escape from predators or to have high energetic activity, for example, chasing down prey items as well. So overall, these metabolic results show us that these fish have higher metabolic demands under pregnancy and have lower maximum metabolic rates under OAH conditions. We also looked at the effects on the mom's blood chemistry. And it's really important for fish to maintain and regulate pH inside of their bodies, because if their pH changes, it can wreak havoc on biochemical reactions and physiological processes. Most fish are able to regulate their pH despite changes in the external environment if there's lower pH. However, what we measured in our experiments was that even under low pH conditions, under low pH conditions, the fish that were exposed to OAH had lower blood pH, yet they were able to somewhat compensate for the effects of low pH by increasing bicarbonate levels in the blood. But nevertheless, the fact is that they still had a lower pH even after increasing bicarbonate levels, suggesting that they could only partially compensate for these changes in pH in the water. In addition, we found that they had higher levels of hematocrit, which is the percent of red blood cells that carry oxygen. And this suggests that they are able to respond to lower levels of oxygen in the water by increasing hematocrit. And so overall, we're finding that the fish are responding to these changes in ocean chemistry. However, they are unable to fully compensate, suggesting that they are going to be physiologically compromised under these conditions. Next, we looked at fecundity and how these OAH treatments impacted the number of larvae that the rockfish ultimately produced during parturition. And so what we're looking at here is a fecundity length relationship. The blue line represents ambient conditions, and the red lines indicate either our high or extreme OAH conditions during pregnancy. And what you can see here is that under OAH conditions, across all size classes, there's a decrease in fecundity, meaning that OAH is reducing the number of babies that these rockfish are producing. The lower fecundity may result from the energetic shifts that we were measuring, the lower maximum metabolic rate, and also energy potentially being diverted away from reproduction and towards acid-base regulation. When we looked at the impacts on larvae, we found a number of concerning traits. So we found that OAH reduced the size of the larvae. It also resulted in smaller otolith area, and the majority of the larvae that were born into these high and extreme OAH conditions were premature. So on the right, I'm showing you what a full-term larvae looks like versus a premature larvae. And we found that in these high and extreme treatments, that the majority, or in the case of the extreme, 100% of the larvae were born premature, which means they're going to be smaller at birth and spend a longer time period in the plankton before they can reach the juvenile stage. And thus, they're much more likely to not survive through the next stage of life if they are born premature. So overall, what we found for this first objective is that the moms had reduced maximum metabolic rate and altered blood chemistry, suggesting physiological impacts to the moms. And the babies had, there were fewer larvae overall with lower fecundity, and those larvae that were born were smaller and developmentally premature. The next question that we looked at was whether maternal OAH exposure could potentially boost offspring performance, with the idea that there might be transgenerational plasticity effects whereby the mom could potentially prime the larvae to deal with similar environmental conditions better because the mom is already experiencing them. And so for this, we did another set of laboratory experiments, again with our focal species, the gopher rockfish. We collected gopher rockfish, again from Monterey Bay, and in this set of experiments, we exposed them just to either ambient or a high treatment from our previous objective that, as you saw, induced OAH impacts. And we varied the timing of exposure so that the rockfish females were either exposed right before they fertilized their eggs or during the gestational period. And for these sets of experiments, we then did a reciprocal larval exposure where the larvae were either then held in the same conditions as their mothers or into the opposite condition for 5 days. And then we looked to see whether or not the larvae performed better if they were held in the same conditions as their mothers. The response variables that we looked at were larval performance and DNA methylation, with DNA methylation being a potential mechanism by which the moms might be able to confer resistance or send a message essentially to their larvae that they might be dealing with similar environmental conditions. And so here's just one of the series of performance indicators that we looked at. This is larval activity. And what we found was that maternal exposure improved larval performance under similar conditions. And this is a little complicated graph, but what you can see here is the maternal condition here, and then the larval condition if they were born either into ambient conditions or then placed into an OAH treatment. And what we're seeing here is for the larvae whose mothers were placed in the ambient treatment, that they had a significant decrease in performance if they were then placed into the OAH treatment. However, we did not see this in the two groups where the maternal treatment for the larvae had experienced OAH conditions, either during gestation or prior to fertilization. In both of these cases, we saw a decline, but we did not see a significant decline, suggesting that potentially the exposure during the maternal period could boost larval performance under a similar OAH treatment in the larval environment. Next, we looked at whether or not we saw any signs of differential methylation. So this is DNA methylation, which is a epigenetic modification that can alter whether different genes are turned on or off. And so we're looking at larval DNA methylation patterns here. These are different genes, and the colors represent either more methylation or less methylation. And the maternal groups are shown here at the bottom: gestational, ambient, or exposure prior to fertilization. And I'm not going to spend too much time on this, but the take-home message is that we did see significant differential DNA methylation in the larvae that depended on the treatment of the mother, suggesting that the maternal environment is influencing gene regulation in the offspring. The next step is for us to then identify what these genes are and how differential regulation of these genes might be impacting performance. And this is a really cool and exciting finding that we're looking forward to digging more into. Summing up this objective, can maternal OAH exposure boost offspring performance? Yes, but it does not totally offset OAH effects, but we are seeing signs that this might be the case. And we're also seeing evidence for maternal buffering via DNA regulation, which can impact gene regulation. Next, we looked at whether real-world ocean acidification and hypoxia variability impacts rockfish reproduction in the field. So to address this objective, we went out and collected rockfish across 2 field seasons during 2023 and 2024. We went fishing every other week pending conditions, and we were interested in looking at the reproductive stage and fecundity of a variety of nearshore abundant spring spawning rockfish species. And we also looked at their hepatosomatic index as a measure of energetic stores that might be that might be impacted by changing oceanographic conditions. So these fishing trips spanned the prior to and during the upwelling season. So we were lucky enough to actually have 2 differing oceanographic years. So we found that the 2024 year was warmer and there was overall weaker upwelling, which you can see on this bottom plot. We have dotted vertical lines representing the dates of our different fishing trips across the 2 seasons. And you can see in 2024, there was a lot less upwelling compared to 2023. And when you look at the El Nino Oceanic Nino Index, you'll see that during 2024, it was also a warmer year with El Nino occurring during the first part of 2024, which coincides with the spring spawning season. And what we found is that there was higher reproductive investment in 2024. So these are 3 different species that we measured. This is weight-specific fecundity, and you can see that in 2024, fecundity was higher across all 3 species. And again, 2024 was the year where we had less upwelling and slightly warmer conditions. And this is expected based on prior studies showing that spring spawning species tend to have higher recruitment during years with lower upwelling and warmer conditions. And so we did see that reproductive investment varied with oceanographic conditions. We also looked at energy storage or energy that was allocated not toward reproduction and towards growth by measuring the hepatosomatic index. And what we found was the exact opposite of what we saw for fecundity, whereby the rockfish were investing more into their liver energy stores in years where reproductive investment was lower. So basically, they're saving their energy and not investing as much in reproduction during those years with potentially less oceanographically favorable conditions for reproduction. We also found that there was a shift in the peak larval release for gopher rockfish. And so what we're seeing here is this red line is the peak larval release in 2023, and the blue line is the peak larval release in 2024. And what I want to highlight here is really just that there was a shift between the 2 years, and that the shift overlapped with the rockfish fishery opening day. So for those fish in 2024, this means that about half of the fish were still pregnant after the fishery opened, which could be problematic and shows that the fishery closure may not be protecting all of those pregnant females, and suggesting potential management changes that could shift during different oceanographic years to better protect these spring spawning species. So does real-world oceanographic OAH variability impact rockfish reproduction in the fields? We found that egg production varied with upwelling intensity across years, and we also saw that the peak larval release varied in our focal species, the gopher rockfish, which may indicate that there's a mismatch with the fishery closure period. And then our last question was, how will future OAH impact rockfish populations? And the idea here was to incorporate some of our laboratory experimental findings into a stock assessment model that would allow us to then project how these changes in ocean chemistry might impact populations to better inform management. And so what we did was, for this objective, we took that the fecundity length relationship that was altered under OAH, and we incorporated that into the existing gopher rockfish stock assessment model that's currently used by NOAA. And what we found is that when we incorporated this new revised fecundity length relationship, it actually improved the ability of the stock assessment model to predict recruitment. So what we're looking at here is a plot showing observed recruitment versus the larval output using the OAH-informed model. And you can see that it explained about 26% of the variation. So this was both sort of unexpected and exciting in that we were able to integrate our laboratory results into this revised stock assessment model, and it helped to explain past recruitment variability. The next step was then to use this revised stock assessment model to make forecasts under future pH and DO conditions. So these are pH and DO for the Central California area from a downscaled ROMS model that was given to us by Jerome Feichter's group at UC Santa Cruz. So we used the revised stock assessment model and applied it to these projections of pH and DO to look at how populations of gopher rockfish would change into the future. And so what we're seeing here now are those future predictions. We have the percent change in total larval output on the y-axis here, and then we can see how that changes from 2000 to 2100. And essentially what we're seeing here is about a 20% reduction in total larval output from the stock assessment model, suggesting pretty significant declines in total larval output in the future. One thing that we can't determine from these results, though, is whether or not it's really pH or DO that's driving these changes, because our laboratory experiments manipulated the two together. And so that's just a caveat to looking at these projections. To sum up, how will OAH impact rockfish populations? We found that our OAH-informed stock assessment model helped to explain additional recruitment variability beyond the current stock assessment model. And we found that the projections suggested a 20% decline in egg production by the year 2100. So I've shared with you a lot of results, and I wanted to now take this back to the potential management implications of this research. So thinking about this call overall, one of the things that we were interested in doing is to protect marine biodiversity and water quality under OAH conditions. And so through our research, we were able to identify how natural OAH fluctuations impact rockfish reproduction. And we also found evidence for maternal buffering, which could contribute to the adaptive capacity of this group and potentially be a means of rapid adaptation to changes in ocean chemistry associated with OAH. In terms of supporting climate-ready fisheries, our group developed this environmentally informed stock assessment model. And our data also highlights how climate-informed rockfish management strategies might be more important for investigating for these spring spawning species, which may respond to different oceanographic conditions in the timing of their parturitions. And then lastly, how have we advanced coastal adaptation efforts? We have produced projections of how rockfish populations might decline in the future. This information can be useful for stakeholders, including the fisheries themselves, as well as for tourism and local jurisdictions. And we've contributed to workforce development through a massive amount of training. We trained 6 master's students who are the ones who primarily implemented and produced the data that I shared with you today. We had more than 30 undergraduates that worked on this project. And in addition, we implemented a K-12 ocean acidification outreach program that hit more than 800 students in the Tri-County area of Monterey, San Benito, and Santa Cruz counties. I want to acknowledge the many, many people that helped to implement these experiments and produce the data that I shared with you today. In particular, the 6 master's students that basically ran the project, 2 of which have now graduated, and the other 4 are still working on their theses. I'd like to acknowledge Neosha and Dave at UC Santa Cruz, who are NOAA-affiliated staff scientists. postdoc collaborator, Dr. Garfield Kwan, as well as our OA outreach lead, Dr. Corin Sloan here at CSU Monterey Bay, the many undergrads, and our scientific illustrator, Evelyn Lamb. And with that, I'm happy to take any questions, and I'll leave you with some photos from our OA outreach program here on the right. Thank you.
Thank you so much, Cheryl. Great presentation. We do only have a couple minutes left for Q&A, so we do have some in the Q&A function. If anyone else has, please add them in. And Cheryl, if if we don't get to them all, you can respond to them via writing to them. But we do have one in the Q&A. Let's see. From another researcher, Alex Harper, asking what were the extreme pH exposure levels and how did you select high pH and extreme conditions?
Great question. Let me see if I can go back to that slide. Okay, so these were the conditions that we used for the first set of experiments. And so we looked at how pH and DO co-vary during upwelling using sensor data that had been previously published. There was a paper by Christy Croker's group a few years ago that looked at how pH and DO vary during strong upwelling events. So that helped to drive how we selected the values of pH and DO that we used. When we got to the extreme conditions, we were a little worried about going too low with the DO because we wanted the rockfish to still have successful parturitions. And so we didn't go any lower than 2.8 milligrams per liter.
Excellent. Thank you. And then we'll have time for one more. Another researcher said, "Great talk. And wants to know is the timing of rockfish peak larval release always tightly timed, or is this less well known? And then not entirely clear whether this is due to the unfavorable OAH conditions."
So I don't think we know the exact timing. I mean, we know that the gestational period is about 3 to 4 weeks, and there's variation within the population as to when they actually fertilize their eggs and when that gestational period occurs. It definitely varied a lot in our laboratory experiments, with some of the fish going maybe a month earlier than other fish. And so there's, I believe, natural variation, but it has been shown in some previous studies that recruitment is later in years where oceanographic conditions are more favorable. So that finding has been seen before, and so it wasn't entirely unexpected that we saw this shift. Although we only have 2 years of data, so we can't really say for sure if it was driven by these exact oceanographic conditions. If we had more years of data, we would have a better, stronger ability to tie it directly to the types of conditions that we measured in these 2 years.
Excellent. Thank you, Cheryl. There was a question for if you could share the K through 12 curriculum in the chat, that would be awesome. And then there are a couple more questions in the Q&A feature. If you wouldn't mind just taking your time to respond to those, as we do need to move on at this time.
Of course.
Thank
And
you.
I would be happy to share more about the ocean acidification outreach program, including the curriculum for anyone who's interested. Thanks again.
Yes, thank you. All right, so we're going to move on to our next speaker, Eric Bjorkstad from Humboldt State University. Feel free to share your screen and take it away.
There we are. All right, can everybody hear me? I hope so. Can someone confirm that they can hear me?
Yes, we hear you.
Yes.
Okay. All right, so thank you. I really appreciate Cheryl going ahead of me and explaining how ocean acidification works in upwelling systems. Saves the slides that I did not put in. So anyway, I'm here to present on behalf of a group of colleagues. Several of us are based up here at Cal Poly Humboldt. Several are from Mbari, Yui Takashita's group. And Central Northern California Ocean Observing System helps with a lot of the data management. Our project is focused on observations and operations around observations. Just to set up the framework here, the problem and the opportunity that kind of motivated this project was looking at California's North Coast being a really under-instrumented, under-resourced region for ocean observations, which means that the communities up here that rely on ocean fisheries and the tribal communities that have strong cultural and economic ties to coastal resources did not have a lot of information about the environmental impacts and environmental dynamics that might affect those resources. It's also interesting coastwide because of the proximity of the Klamath River and the Chinook salmon fishery here that are a linchpin for West Coast fishery management. And it turns out that the North Coast up here is a hotspot for both OAH and for HABs. Part of the motivation for this project was we have this long-running ocean observing program up here, the Trinidad headline. And when we looked at OAH and HAB events resolved on a monthly cruise timescale, we noticed that there was some correlation, some suggestion that these dynamics offshore were in fact connected to some of the variability we were seeing onshore at the Trinidad Pier shore station as part of the Senku shore station network. These two plots here to the, let's see if I can actually get a pointer here. These two plots here show variability in pH over the course of some of our cruises relative to the higher variability at pH at Trinidad Pier, and likewise for DO here. And so project goals here were to augment OAH observations on the Trinidad headline on our ship-based work, possibly extend sampling into state waters. Core goal was to establish what we now call the Trinidad Head Ocean Observing Node to monitor ocean acidification, hypoxia, et cetera, and other oceanographic parameters at high temporal and vertical resolution at a mid-shelf station. Start to bring these data streams into possibly into forecast models, or at least understanding what risk might be affecting shelf and nearshore waters, and then to build a high-quality data set to support this kind of analysis and provide a framework for validating some of the modeling that's been developed in this area. Here's the realized project goals, because we're doing operations in a challenging environment, and it's certainly a challenging environment with respect to, well, I'll get into that in a second. Some of the things that we have not done is extending the sampling in. We did make an attempt at that, and that we nearly ran our CTD into a rock pile, which would have been a catastrophic loss. So we cut that out and didn't waste our time in there in the dangerous spots. We are still working on the analysis of the data that has emerged, but we continue to carry on. So the Trinidad Head Ocean Observing Node, it's co-located with what we call station TH02 of the Trinidad headline, which we see here, which happens to be a focal station for an ecological copepod-based time series that we collaborate with the Newport line up in Oregon on understanding. It's a suitable or useful indicator for looking at salmon dynamics and ecosystem state. The station is it's situated mid-shelf. It's about 10 kilometers offshore, sits in about 75, 80 meters depth of water, and consists of two co-located elements. One is a wire walker profiling mooring, and the other is a seafloor mooring, which I will describe for you now. So the profiling mooring is based on the wire walker platform. And this consists of a surface buoy from which depends a weighted cable, and a wire walker sits on this cable, uses a cammed ratcheting system to use the undulation of the buoy and its cable, and the inertia and friction that the wire walker itself has to climb itself down the wire. When it hits the wire, the cam releases, and we get a nice smooth ascent along the cable, during which the upward facing sensors get a nice clean look at what the water column looks like. And under normal wave conditions up here off the North Coast, this excursion from about 70 meters depth to very near the surface, 3 to 5 meters, occurs every 8 minutes when the system's in operation. Now, one of this project was ambitious in two regards. One is it was one of the first and coincident implementations of an experimental MFET-based pH sensor that Yui Takashita's group has developed, integrating that into a wire walker, which required mounting a second battery pack, the sensor and its pump system, et cetera, into this limited space, and making sure that we could get enough ballasting foam in there so that the ascent would be at the correct rate and that it actually would ascend. That's also important. And then also the endurance of the deployments that we tried was well in excess of what the wire walker was typically designed for. We were pushing for month to 2 month long deployments. The wire walker was initially designed to be used for process-based experiments that were more limited in duration. The seafloor mooring consists of an upward-looking ADCP to resolve currents throughout the water column from about 70 meters depth up to as close to the surface as we can get based on sea state and noise and reflections. That will give us a sense of which way the water is moving over Thun and through Thun. And it also carries a deep sea fox to measure temperature, salinity, dissolved oxygen, and pH in the bottom boundary layer, very near the sea floor. This is what it looks like kind of vertically. We have this tethered then to a SOFAR spotter buoy that provides some ancillary information on waves and sea surface temperature, but also provides us a very easy way to recover this when we do the mechanical release. We can know that it's there because the SOFAR buoy is telling us that everything's okay. This piece of Thun is modeled on work that's been done up off Oregon in Jack Barth's group, looking at hypoxia dynamics up off the Oregon shelf. So here gives a history. This is these are curtain plots of temperature by depth over time, coupled with plots of pH in the red and dissolved oxygen concentrations in the teal or gray. For the 3 years so far that we have deployed the instruments, 2023, 2024, and 2025. This is the history of the data. We obviously have other parameters. I'll show you those in a minute. This has been a challenging project. Suffered delays in terms of delivery of equipment, tariff-related delays in getting equipment returned back across the border from the calibration from the manufacturer. When we started pushing the endurance of the wire walker and its sensors, we realized that we were producing data faster than the wire walker could communicate it. So we get some of these discontinuities. We had a lot of work with the program engineers to try to resolve this comms problem. Disruptions and disappointments relate to both Jacob Partita, the technician, and I suffered career disruptions, so to say, with our federal employment that really impacted 2025. But we also have data, and more importantly, we have designs, because we are currently queuing up with some additional support from Senkus, et cetera, to deploy in 2026, and we're looking good for an April deployment. The instruments look great, so we're hoping to get out there and have a nice long season. So what do the data look like? Co-located time series of temperature from the top down, temperature, salinity, and calculate density from that. Dissolved oxygen, pH from this experimental sensor, which is really cool because it's resolving this at high resolution in a profiling mode. This is a fairly novel technology in the pH world. At least it certainly was when we were heading out to do this. And then coupling it with chlorophyll. As I said, we have here on the top, just for context, these are the winds coming out of the north, and these are the tides. Profiles from 70 meters to about 3 meters every 8 minutes when the system is operating. This is a span of about 10 days in June of 2023. A couple of the products that we have been using, Senkus has been very integral, Maureen Lebrecht and Fred Barr especially, and helping us, one, to build an internal Wirewalker QC dashboard. This has been really important for being able to monitor the state of the instruments, to support UE's team in understanding the performance of their new pH sensor and dialing it in. There was a lot of R&D kind of work, especially in years 1 and 2 of the project. But we also take the data sets that we are returning. The Wirewalker and the SOFAR buoy, both of those data streams come to land in near real time. They're telemetered by cellular networks. And we took those data and created a portal or a little website that served things like significant wave height, temperature at the surface and at the seafloor. It also, this is vertically truncated. It also has dissolved oxygen and chlorophyll, both at the surface and at the seafloor. And we put this together partly to advertise that Thun was out there. Please don't tie your boats up to it, but also to provide real-time information to local ocean users. This mooring sits out in some of the prime Pacific halibut fishing grounds. And so it was something that would be of real-time interest to the ocean community. So back to what we hope to learn, well, what we can resolve and what we hope to bring some science to light around are the dynamics that we're able to observe with this instrument. We can see a mid-shelf response to upwelling. We see the upwelling here, cold water relaxation as the winds abate here, and how the mid-shelf water column responds to that, or the water column that sweeps over Thun responds to that. We see evidence of stratification, some of this density compensation between temperature and salinity that's fairly typical of a front passing by. There being phytoplankton blooms, corresponding changes in dissolved oxygen hypoxia, and of course tracking what's happening at the bottom. Really neat, we can actually see internal tides. This is tides at North Spit up here, and you can see these pulses. These are actual internal tides coming in, breaking over the shelf break and driving substantial vertical excursions in all of the isoplex for temperature, all the quantities. You can kind of see this dynamic happening. When we look at longer time series, I can't remember if I have this slide, and some of these big excursions actually show up and propagate all the way to the bottom. So you see high variability in oxygen and pH in the bottom boundary layer, driven by these tides. So one of the big questions, and this is where we're kind of leaning to take the data and the analysis that motivated the project, was, what is the connection of mid-shelf to the coast? On this left plot, we're showing here that pH and dissolved oxygen resolve to integrated winds. These are winds integrated using a method pioneered or discovered or established by Austin and Barth to kind of characterizes the coastal ocean's response to upwelling favorable winds and how long that inertia persists. So it's an integrated wind measurement. And we see that when the winds out of the north are stronger, we tend to have lower pH, both at the shelf and also showing up at the coast. But there's a huge, as you can see in these time series, huge amount of variability, certainly at the coast, at the Trinidad Pier Shore Station. What's nice is we can see that the evidence here that the observations taken at shelf waters in real time, these are things that we can return with the Wirewalker, provide a worst case scenario of what the source waters look like that could be delivered to the coast during an intense upwelling event and how that would change. The correlations on a day-to-day scale between the shore station and the offshore bottom waters is not very strong. So modeling will have to take into account this kind of conditioning of the water, but also account for the fact that coastal waters are not tightly coupled to the shelf. Intense upwelling events certainly can drive that coupling, but when that intense upwelling is not active or there's more variability, we see this isolation or this decoupling between the coast and the shelf, and a lot of biological modification in terms of drawdown of pH and increase in dissolved oxygen. You can see that especially on a day-to-day scale at the shore station. So to kind of frame some of our preliminary conclusions and how we're looking ahead, I'm going to answer the question that was posed to the presenters. And, how does your research provide management agencies and local jurisdictions with information to address a number of management issues that you can see here? And I think three things are kind of coming out of this, and will continue to come out of this as we continue to operate Thune as part of Senkus and possibly as part of other research and monitoring programs. One, we're providing direct observations of ocean acidification and hypoxia over productive shelf habitats. These are habitats that are occupied by Dungeness crab, various rockfishes, lingcod, as I mentioned, Pacific halibut. The idea of if this had been out there in 2015, we would have caught the hypoxia event that occurred, briefly occurred over the shelf during the peak of the harmful algal bloom, for instance. As I mentioned, it is information on what's the worst case exposure scenario for coastal waters. Have some potential for early warning based on state of waters over the shelf. We're going to continue to integrate this with some event modeling that was developed in as part of the motivation for this, and has continued to evolve. And this is another, I think, forward-looking value of this project and this investment in observing technology is that these are high-resolution observations throughout the water column. They're returned in near real time. This is the kind of information that is, I think, going to be really valuable as we start to try to understand better drivers of what generates and sustains harmful algal blooms, possibly supporting the modeling around HAB forecasting and validating some of the biogeochemical aspects of some of the models. This is something that we go by the ship all the time, and we're collecting DA samples, things like that. We can, using this allows potentially even adaptive responses for enhanced sampling, because we can see what's happening and how it's evolving and how it's happening throughout the water column, not just at the surface. And with that, I just wanted to really quickly remind everyone of, Yui and his team, Brent and Joseph especially, and Irene as well, really helped with dialing in the pH sensor and supporting that from afar as we were getting that configured. Maureen Lebrecq was really instrumental during her time at Senkus in getting our portals and QC websites stood up. And Jacob Partita has been the lion of this project in terms of getting the instruments together, configuring them, working with the technical folks at Del Mar Oceanographic and RBR to get the instruments configured into the wire walker, working, communicating with each other. He deserves a lot of credit for making this happen. And with that, I'd like to thank you for your time and invite any questions.
Thank you so much, Eric. We do have a question, but just to notify everyone else, if you do have any questions for Eric, please add them into the Q&A feature. You might have to click the More button on your Zoom bar. But one question did come in from a fellow researcher here today, saying, amazing data, Eric, for the pH sensors. Is there a best speed for the wire walker to profile? Can it be too fast for the pH sensor?
So great question. So the wire walker is ideally that's actually a question that affects all of the sensors. All right. So and the wire walker is ballasted so that on a free ascent, it ascends at about 0.3 to 0.4 meters per second. And which matches the response time for, say, the dissolved oxygen sensor and the obviously temperature and conductivity sensors. The engineering around the MFET sensor was specifically focused at tuning that element for profiling applications. They are now being put on gliders. The Embari team had a wire walker themselves that they were working on in parallel with us. We actually had a coupled project here. So it's tuned to do pretty well at that speed. And the high correlation, didn't show a correlation thing, but the high correlation between the dissolved oxygen and the pH sensor, which we would expect to see, kind of helps corroborate that. but it performs pretty well. If we had it coming up at a meter per second, probably a problem. You start getting into that. You overrun the sensitivity or the response time of the sensor somewhere in that range. It is also, I should say, it's a pumped sensor, so it actually is pulling water in at a controlled rate across the sensor.
Great. Thank you, Eric. Are there any questions? Oh, but we just got another one. Another fellow researcher has asked, would one of these Wirewalker mooring work at, say, the mouth of San Francisco Bay, just beyond the Golden Gate Bridge, to better constrain ocean estuary flux?
Yeah, I think it would. The challenge there is just making sure that it is robust to the currents, the tidal currents. The Wirewalkers are themselves designed the way they're configured to keep that cable below the main mooring for it to stay vertical, so that the Wirewalker package itself will ascend smoothly. But so long as those logistics can be worked out, it would be great. I think this technology has a lot of potential for coastal observing. The quality and the resolution of the data is pretty amazing. So.
Excellent. Another question came in. Is there a desire to be able to control the ascend rate for the Wirewalker? And this is from just an attendee today.
So the Wirewalker ascent rate is, if you will, controlled in that we tune it to be in that range by putting in different amounts of ballasting. And that's not something I think you would want to vary a lot, because that is tuned in order to collect quality data throughout the ascent by letting the sensors come quickly to equilibrium or respond quickly to the conditions as they change coming up. So that 0.25 to 0.5 meters is kind of the sweet spot for the Wirewalker. 0.5 meters to a meter is typical descent rates for CTDs, for the oceanographic sensors that we send down off ships. So that rate is about that's pretty standard across sampling platform sampling techniques. If you wanted it to be, let me. If it's, we also, I should say too, and this, I don't know if this is what the questioner was asking, is the interval between profiles is entirely dependent on the wave climate. If we're in an really energetic wave climate, we might get profiles every 4 or 5 minutes. And then when it's really calm, whoops, lights just went off. When it's really calm, they may be separated by 10 or 12 minutes, because the intervals that make the Wirewalker go down just slow down. So it is dependent on wave climate.
Excellent. Well, thank you, Eric. I think that's all the time we have for questions. If any more come through, you can find them in the Q&A feature, and you can just type your answers to them. And Cheryl, I'll just say that settings have been changed. You should be able to see those questions now, because I know there's a couple in there for you still. But with that, we can go ahead and move to our next speaker. We have Christina Frieder from SCORP going to share their research with us. So take it away, Christina.
All right, thank you. Yeah, that was really, this is really great to go after both Cheryl, who introduced OAH so well and why it matters to marine organisms, particularly those that require oxygen to breathe. And then also Eric's talk, who really demonstrated all of the chemistry data that we're starting to amass in terms of showing ocean acidification and hypoxia patterns along the California coastline. So I think that this will be a nice bridge to my talk about developing multi-stressor tools that help us interpret the effects of acidification, hypoxia, and warming for California's marine organisms. I'm presenting this on behalf of my coworkers at SQUIRC. That's the Southern California Coastal Water Research Project. And this is in partnership with Curtis Deutsch, who's at Princeton University. And of course, thank you to OPC and Sea Grant for the funding to do this project. Yeah, so I already mentioned Eric had a really nice demonstration about the increasing amount of observations we have in respect to pH and oxygen data. Not only do we have increasing amounts of observations, but we also are having increasing amounts of model outputs that are demonstrating changes since before the pre-industrial revolution, so since before we started emitting lots of greenhouse gases to our atmosphere and the oceans started absorbing that excess carbon dioxide. But we're also getting model scenarios about future pH trends anticipated into the future and how that varies under different emission scenarios. And something really unique along the California coast that both of the other projects really highlighted is that pH and oxygen really co-vary along our coastline. And so the biological responses to those exposures aren't just in response to low pH alone or low oxygen alone, but really coupled together. And of course, temperature is varying with these as well. I just didn't plot it on this plot. I kept it in 2 dimensions, but we also know that, that temperature co-varies with oxygen and pH as well. But where we really are lacking is a mechanistic framework that allows us to interpret, say, time series or model outputs of pH, oxygen, and temperature to the biological responses. We do have some biological linkage tools that are already in application that I just want to demonstrate here. We have single stressor biological interpretation tools. Here's an example on the left of using ocean acidification thresholds. So here's an example of looking at, in the Southern California Bight, what is the typical amount of habitat where aragonite saturation state exceeds 1.4. We know that this value is important for pelagic calcifiers, particularly pteropods, in their physiological performance and their ability to calcify their shells. So this is showing gradients in that habitat thickness with respect to aragonite saturation state. And this is from model outputs in the Southern California Bight. I'll then contrast that with, on the right, with an interpretation, a mechanistic interpretation that includes both changes in oxygen and temperature to talk about aerobic habitat availability. So the one on the right is showing the amount of the water column that is has sufficient oxygen at a given temperature to be aerobically available for a given marine taxa. I don't have it here, it's what species this is, but this was specifically for northern anchovy. So this is just an example about how these sorts of biological linkage tools really help us interpret observational data and model output data to talk about when is it that changes in chemistry matter for changes in biology. But you'll see that these are currently independent. We have OA on one side and oxygen and temperature on another side, and we don't have a mechanistic framework that combines all 3 of these, both OA, oxygen, and temperature all into the same one. I want to answer that question about how this is important for resource agencies in terms of helping them make decisions or manage our resources. And these types of biological linkage tools have been used or are helpful to resource agencies to really evaluate when changes in water quality are translating to biological effects. So here's an example of using these biological linkage tools to look at the effects of land-based nutrient inputs into the Southern California Bight. we have both natural upwelling that brings nutrients and causes phytoplankton production, but especially in the Southern California Bight, which supports a population of over 20 million people, there are also substantial amounts of nutrients that are entering our coastal ocean from the human-based sources. And here's showing the changes in aerobic habitat thickness. So the oxygen temperature space that is available to northern anchovy in this case, it is showing those changes when you include land-based nutrient inputs and the downstream effects that has on oxygen in the Southern California Bight. So you can see that the red areas are showing areas of habitat compression versus the blue areas showing aerobic habitat expansion. We can also use these sorts of tools about protecting biodiversity and understanding changes into the future. So another way we use these biological linkage tools is with climate change scenarios and thinking about future climate emissions pathways and how habitat and availability of habitat and those biological effects are going to change into the future. So how are we going to come up with an interpretation framework for our observational data and our model outputs that integrates oxygen, pH, and temperature? And for this project, our starting point was to begin with the metabolic index. We really favored the metabolic index because it is mechanistic. It's already a multi-stressor index in that it incorporates both oxygen and temperature information. And so we just have to expand it to also include pH as well. It's also been applied across a range of global marine taxa or across the global ocean for many marine taxa. And there are now metabolic index traits available for over thousands of marine species. And so it's been shown to be a really robust framework about thinking about interpreting how the conditions of the marine environment are available to a given species. So this was our starting point to think about expansion of the metabolic index to now include not just oxygen and temperature effects on aerobic habitat availability, but also the influence of pH. So the question is, how can pH modify the metabolic index? This is just sort of a conceptual for a range of ways, but the metabolic index is really a ratio of an organism's oxygen supply. So you could consider how pH modifies oxygen supply in an organism. And Cheryl had some great examples about changes in blood chemistry and that could actually be influencing their efficiency to supply oxygen to their tissues. And then that's a ratio over an organism's oxygen demand. And there are many cases in the ocean acidification literature showing how metabolic rate can change with it can either decrease under ocean acidification scenarios. It can either be unaffected by ocean acidification scenarios, or here on the left, I'm just showing how you can have an increase in metabolic demand of a given species under an ocean acidification scenario. And we're even starting to get a better understanding about what's happening within their energy requirements that causes those shifts, either the increase in energy demand, no change in energy demand, or a decrease in energy demand. So our approach for this project to expand the metabolic index to include pH as a factor within its framework was first we conducted experiments that measure the hypoxia tolerance of Dungeness crab juveniles. And we did that in response to both a range of temperature conditions as well as pH conditions. That was just for a single taxa, but then we also performed a literature review, really focusing on the ocean acidification literature, which has been amassing over the past 2 decades to look at how metabolic traits respond to ocean acidification and temperature, to look for, is there universality in the response, or can we categorize it somehow in a way that allows it for simple adaptation into the metabolic index framework. Our third task was then to, from these findings of 1 and 2, to incorporate ocean acidification into the metabolic index framework, to talk about aerobic habitat for marine organisms. And then we apply those biological linkage tools to some ocean model outputs that are available for California's coastline, to look at the relative importance of oxygen, temperature, and pH. So here are just some, I'm going to share results from our experiments. We did experiments, again, on Dungeness crab juveniles, where we measure 2 aspects of their metabolism that are particularly important in defining their metabolic index. And those metabolic traits are, first, the critical oxygen pressure. So that's the graph on the left. This is essentially the oxygen value at which an organism can no longer maintain its metabolism. That's essentially saying there is no longer enough oxygen in the environment for them to maintain their basal metabolic needs. So typically you see them die, or you see organisms die or begin to fail when they get to values below these critical oxygen pressures. That graph on the left, you can see that the P-crit, critical oxygen pressure, scales with temperature. And this is actually a very typical fashion, a very typical outcome as it relates to other marine taxa as well. We also measured metabolic rates, which is just what are the juvenile crabs' metabolic rates across a range of temperatures. And again, metabolic rate scales with temperature. And it does it in a fashion that's very similar to other marine taxa as well. The next question was, okay, is there an effect of pH on these 2 response measurements? So the critical oxygen pressure and the metabolic rate. And for these juvenile crabs, we found that there is no effect across 2 treatments. We had a control pH of approximately 8.0, and then our low pH treatment, which was about 7.7, but saw no effect on either of these response metrics. So this would indicate that when we add pH into the metabolic index, the fit for Dungeness crab, when you do it specifically for Dungeness crab, that there's no sensitivity of the metabolic index to varying pH or including pH. But that's not necessarily the case across all marine taxa. In reviewing the ocean acidification literature, we already know that there's a range of responses in metabolic traits when you change pH conditions. So here's just 3 example species that all have different responses, what we would categorize as a negative effect of pH on metabolic rate. On the left, that's a pteropod species. In the middle, you have a bivalve species that really has no effect. That's similar to what we saw for the Dungeness crab. And then you have species that fall into this positive category where there's an increase in metabolic rate with increasing pH. And so our literature review to date does find that that species range from this negative to positive effect of ocean acidification on metabolic rates, and that there are many species that fall close to the neutral range, so their metabolic rate is largely unaffected by pH treatments. But you do have some species that are highly sensitive in either direction. And so this literature review is underway, but we've identified 36 species so far that had been studied in an experimental lab setting. And so this is something that we're still working to expand. But this x-axis right here, the slope, is then how we're going, meaning the extent to which this species is sensitive to pH, is how it's going to get incorporated into the metabolic index. So this is the full equation for the metabolic index. You can see I highlighted in red that it already includes oxygen and temperature. But now we have a way to include pH as well and the sensitivity of a species of its metabolic rate to pH. So we have a mathematical way to expand the metabolic index for applying to observational data and to model outputs. So really moving forward, our last step is then to do some test studies for a couple taxa to look at what is the relative role of oxygen, pH, and temperature in constraining habitat availability, both now over the past few decades, and then also looking into the future under contrasting emission scenarios. And this work is really in progress. I've shown you that we have our literature review. We are expanding. We have a mathematical way for which to expand the metabolic index, and so we are actively now applying this to model outputs to answer this question. But I want to just give you an example of using the metabolic index from northern anchovy before this work, where it's just dependent on oxygen and temperature. And we know that in the Southern California Bight that there is seasonality and the amount of aerobic habitat available for northern anchovy, that being constrained by oxygen and temperature conditions. And when you look at the seasonality and attribute how are these changes driven by changes in oxygen in the Southern California Bight versus changes in temperature in the Southern California Bight, we know that the seasonality and habitat compression that happens in late summer into early fall is entirely driven by oxygen. And in fact, the temperature counteracts that which is driven by oxygen to a small extent. So this is one example of how we've concluded that oxygen really drives aerobic habitat availability on a seasonal basis in the Southern California Bight. So next we're going to be doing this with a pH-dependent metabolic index to look at their relative contribution now on different space and time scales and into the future. So with that, I'd be happy to take any questions.
Awesome. Thank you so much, Christina. I'm not seeing any questions coming into the Q&A yet, but if anyone does have any questions, please go ahead and add that.
I'll ask a question. Maybe this was outside of scope of the project, but was shelled or carapastal solution measured at all for D-crab lab experiments?
Oh, no. So any aspects of dissolution wasn't included in these studies. These are, for these assays, they're exposed to different temperatures, and we hold them in bottles where we measure changes in oxygen over time. So that's kind of how the design of these experiments, yeah.
Oh, sorry, that was a brain fart then.
Yeah.
Anyone else have any questions for Christina? And that's totally fine. But if you do, please add them to the Q&A. And Christina, if you just keep your eye on that, you can see if any come in for you and you respond to them. And if we have any time at the end, we can always come back. So thank you. We'll move on to our final speaker. We have Alex Harper from Sencus and California State Polytech University, Humboldt.
Thank you, Tanya. Can you hear me okay?
Yes.
Excuse me as I clear my throat. And my slides are not in presentation mode. Let me look good.
There they go. Yep, perfect.
Okay, great. Well, thanks so much for the opportunity to present this work. And it's really been fun project and a collaborative one, pulling across a bunch of different groups. I'm happy to go at the tail end of the series of talks now that everyone's exhausted your thinking capacity, because the idea somewhat with this work is to reduce the labor around thinking and interpreting, and really provide some easy to use visualizations and interpretations around some of the data and information being generated by programs that you just heard about and many others across the state and the West Coast. And so here's our project team on the right, a collaborative group with strong representation from the observing systems of Southern California. That's Clarissa and Megan. Central and Northern California Ocean Observing System. Myself, Marine. Jan Newton is on the project from the Northwest Association of Networked Observing Systems. Aaron Satterthwaite, similarly from Cal Coffee, a long-standing and critical observing program. And Christina and Fakal, you just heard from Christina from SCORP. And so the point of this work is pretty straightforward: to build the centralized information access and visualization hub, the California OAH portal, to provide automated, high quality, and interoperable data and synthesis products. And the primary audience for this work is written in our proposal is state managers, specifically Ocean Protection Council, Fish and Wildlife, State Water Resource Control Boards. But in going through the process of developing products and tools for managers, we also identify and eventually develop tools for many others, including, importantly, fisheries and shell fisheries, local water quality managers, and local communities generally, who can access now ocean acidification and hypoxia information somewhat at their fingertips. And so we worked across these 4 broad objectives, recognizing this data quality and interoperability challenge was a big one, streamlining and enhancing visualizations, establishing this hub, and then supporting the development of automated and other synthesis and indicator products. And so we were able to launch this OA portal, and I encourage you all to visit it. It's oah.calus.org. We launched a beta version back in 2023. This was largely enabled this sort of quick launch because it builds on the CalOOS portal, the California Ocean Observing System portal, and takes advantage of many of the existing functionalities and tools of the CalOOS portal. And we were also able to sort of bring in quickly some of the existing data sets we had in the broader portal into this OAA focused tool. And so building on this leveraging of existing investment and capacity from the state of California and also from NOAA and the Integrated Ocean Observing System, and just flagging here quickly that this really built on almost 2 decades of investment and work from folks, some of whom you've just heard from, and from across our many esteemed marine institutes, and starting back with the somewhat with the mortality event of around 2009, perhaps before that, and the kickoff of this integrated exercise of bringing folks together to try to solve societal challenges posed by ocean acidification, hypoxia. We participated in this monitoring inventory back in 2017. I helped to lead that as a Knaus Fellow in the OA program, and all the way to where we are, which is really going after the data that we know about, importantly, the chemical observations, but also the co-located biological information to build a more holistic understanding of impacts across the coastline. And so we were asked, and you've heard this from others, around how this work serves to protect California resources. And we think we've accomplished this in a number of ways. And I promise you my slides are not text heavy going forward, but here is one that is, and I'll just flag a couple of things. This real-time monitoring information sort of at your fingertips. This allows state managers and anyone interested to go in and have a look at what the pH or DO dissolved oxygen is across these more populous embayments and regions right now. And importantly, sort of moving over to this resilient fisheries and shell fisheries point, that this is served up also to shellfish growers and fishermen, and helps them to make informed operational decisions around timing at their hatcheries, or where to set a crab pot to avoid exposure to hypoxic conditions and so forth. And so we think this is working to build resilience across a number of important communities. And I'll come back to many of these points throughout the talk. I didn't originally include this slide, but I wanted to flag here, we, as part of this project, were able to host a California OAH symposium at MBARI back in 2023. Many of you attended, participated. We had talks from the State Water Resources Control Board, from stakeholders, from OPC. We had a whole second day on data quality improvements and recommendations, pulling in folks from the NERS and from the National Estuary Programs. Looking back on it now, this was sort of a great event, and I feel like we as an OA community could benefit from another one of these to sort of take stock on where we've been able to improve over the last many years, and what's coming up next. During that symposium, we heard from Justine at the Ocean Protection Council about some of the OA priorities, and particularly this ocean health scorecard. And so we knew we wanted to do our work to make sure that these indicators were accessible, relevant for folks in California, and automated to the best of the ability. And so this gave us some marching orders. We also heard from the State Water Resource Control Board about this upcoming call solicitation for environmental data to support their 303D development process and condition assessment in 2028. And so we worked really hard over the last couple of years to respond to this solicitation, incorporating some of our Senku's shore station data into this solicitation, and this was a rigorous process, and we have some ideas on how to streamline and improve this going forward. As mentioned, data quality is always top of mind for us at the Observing System, and really to note that a lot of the work we conducted on this project was sort of behind the scenes, and sort of understanding where the data were, applying quality control flags and procedures, and then working to get data into public servers, public access servers, in some cases ERDAP or Data One, bringing it into our portal visualization system, and then ultimately working to archive data, and we archive with NCEI where necessary. Much of the data was sort of already accessed. One thing that did come up in that original symposia that I mentioned was the need for additional QC sort of procedures, particularly for the sensor-borne data from our shore stations. And so we've been able to secure some federal funding to spin up project with folks down at Scripps, which will be starting this summer. Okay, here's some of the feature data that's come into the portal. Some of this was in hand, as mentioned, like the satellite data. Much of the model data, the ROMS NEMRO and the ROMS back model data has come in specifically with the support from this project. Much, if not all, of the survey data listed here was enabled by the funding from OPC and Sea Grant, and excited about. And then the co-location of some of this biological data, again, some of the HAB monitoring data we already had in hand. I think krill came in more recently, enabled through this work, the West Coast OA survey, pteropod data. And then there's an activity ongoing, and luckily, we're able to manage some of this with the ongoing support from the Integrated Ocean Observing System at NOAA, that we're continuing to work towards the data integration. So this includes Cal Coffee CTD time series, and I know the Cal Coffee folks are working hard to get this data into ERDAP, and maybe there's an update on that I'm missing. The Mbari OA1 and 2 moorings we're really excited about, and we've been working hard at my program at Suncoast to get this data cleaned up and into the portal. And the ambition there is to incorporate this then into the mooring analysis for the West Coast OA indicator work to sort of fill a spatial gap in that West Coast indicator. Okay, as mentioned, we have established these real-time OAH dashboards to provide real-time conditions with an outlook from the past 2 weeks. We also did some extra work with the Sea Grant folks down out of Cal Poly Slough and Morro Bay and shellfish growers down there to develop these oyster grower dashboards. These were actually originally developed, I don't know, a decade ago by some folks at Cal Poly Humboldt, previously HSU, these oyster dashboards, and with the Hog Island folks. So now we've sort of redeveloped these and importantly included a trend line over the last 2 weeks. This was something important to them. We do have ideas on how to improve these oyster dashboards, including the inclusion of rainfall and freshwater contributions to local nearshore conditions. This seems to be important to the oysters. We've done some work on moorings, and so the 2 NOAA buoys that are in California, CCE 1 and 2, both set off of Point Conception. We worked with the mooring team to develop this dashboard, which can display both moorings on the same plot. And so the CCE1, that's the nearshore mooring, that's in green, and you see some heightened variability related to seasonality and diurnal influences perhaps. And then the CCE2, which is more offshore, gives you that more consistent pH reading across time. And then, as you heard from Eric, we collaborated on the Trinidad Head Wirewalker mooring to develop these dashboards. One is a web app and the other is this QC Dash, but all pathfinding work that will hopefully be useful down the line as this type of gap filling work continues. We work closely with Dick Feely and the group out of PMEL to integrate the West Coast OA cruise data. Here we utilized existing functionality in the portal that we use for our glider data to visualize our glider transects. And so gliders are these offshore, these ocean-going, autonomously operated, torpedo-looking instruments. And anyway, here we're using them to visualize what is this? Underway CO2 data. And you see in the bottom right, you can zoom around in this 3D setting to sort of see where you are in space and time and along the coastal bathymetry, which gives you a sense of where you are along the coast. And then you can plot that again with the pteropod data over on the left. You're seeing pteropod with type 1 or 2 shell dissolution damage, and so starting to get towards this integrated view of OA conditions and where they're occurring alongside these biological impacts. We now have a really large holding of CO2, the partial pressure of CO2 data in the portal, partly through this MoCA synthesis data set, which I'll talk about in just a second, partly from the West Coast OA surveys underway. And then importantly, Embari also has a significant time series of PCO2 underway from their monthly surveys. And so we've worked with Francisco Chavez's team to sort of automate the generation of this indicator plot in the top right. And so this is the partial pressure of CO2 at Monterey Bay, and B plotted against the Mauna Loa, sort of as like a global indicator of CO2 levels, and you could see the slope, the orange slope from the Monterey Bay. CO2 is slightly steeper than that of the sort of global background CO2. And so starting to think about a sort of simple indicator for the state or others, the PCO2 measurements are giving a clearer picture than often than pH, which can be a bit of a mess. DIC and alkalinity, which are expensive and just take a long time to analyze. This underway CO2 data is more near real time. Okay, as mentioned, this MoCA data set, the multi-stressor observations of coastal hypoxia and acidification. This was something that we were aware of when developing the plan for the portal, but we weren't quite sure how we were going to be able to incorporate MoCA into the portal. So I'm pretty pleased that we've brought in all 20 million observations. Importantly, this synthesis product applies a secondary layer of QC on all public access OAH information. This work was spearheaded by Tessa Hill and her esteemed colleagues out of UC Davis, and so we've worked with them on some visualizations. Importantly, MoCA provides access to chemical data from a number of key observing programs. Here, the scroll overs just zoom to Cal Coffee data, historical data. There's also access data. And so again, this data is all integrated into more of a one-stop shop data visualization layer. And again, highly QC'd. Another big achievement of this group and this project was bringing in the Rahm-Spec model that the folks down at Squirt and their collaborators helped to deliver. And so this is work that we wanted to do as part of our sort of 5-year work plan out of Senkus and the Southern Observing System. We could never figure out how to get it going. And so thrilled to have the opportunity and the support from the state to bring this this really important biogeochemical model layer into a form where you can sort of further explore and discover both trends and changes over time. And so the figure in the top left, this is what the model layer looks like. This is aragonite saturation, I think at 100 meters. And so importantly, you can explore various depths, I think down to 1,000 meters. Yeah, this is 100 meters. I see it in the text now. And then and exploring the data, you can utilize this virtual sensor tool. This is something that the Axiom folks have developed for some of our model and satellite data. And so you can pick a point anywhere on this modeled layer and drop a virtual sensor and you'll get a time series of information. Importantly, you can drop the sensor at a specific depth. And then here is generated an anomaly plot of aragonite saturation at this specific point. Pretty fast, excuse me, fascinating stuff. And then over on the right, we've used this virtual sensor tool. You see these points at 1, 2, and 3 in these sort of bioregions or localities to do some comparison using the Rahm-Spec model layer. And that's what you see in this plot here. This is all generated inside the portal. and you can sort of see the change over time experienced at Point Arena is a bit, higher than the other regions. And so, importantly, the OAH portal is a place for you to come and explore and learn. It's not intended to be an analytical tool. We do have friends, however, who are working on the analytics and really digging deep into changes over time. And so this is work that Jan Newton's been leading with colleagues out of NOAA and PMEL, and importantly with Christina Frieder to make sure that California is well represented in this West Coast wide indicator work, and now this California indicator work. I'm not going to spend a lot of time on this slide here. I do have a QR code for a webinar we put on back in September of 25, where Jan will provide a deep dive, and Christina, into this work. But it's to say that, we're working in lockstep with the Ocean Protection Council, with the Ocean Science Trust about the development of these statewide indicators, and intend to do the work to make sure that this effort is automated and available through the CalOOS portal once finalized. Okay, last couple of fun deliverables. This is a report coming out of UCLA and under the guidance of Mark Gold when he was with NRDC. And this was an analysis that we helped with. It includes a discussion of the CEDAN submission process that we had to go through and some recommendations for improvement. It also includes a series of heat maps that we developed using the ROM spec in this newly available form. There's a Cal Coffee biological indicator that was developed as part of this work under the leadership and guidance of Aaron Satterthwaite, who's on the project and I think on the line, and showing some exciting preliminary results around specific species of zooplankton and OA response. And there's a capstone project that Todd Martz was a part of, and some others out of UC Santa Barbara. There's more information on that. Importantly, we've got an important Cal Coffee zooplankton biovolume data set now available in the portal to do some exploration sort of live. And so in conclusion, some unsolicited recommendations. I'm not going to read through them all here, but it's the idea is sort of to do more. We want to maintain the OA portal. We want to continue to improve it and see it updated. Similarly, with things like the MoCA synthesis data set and ROMspec, we'd like to see those continue to be updated routinely and made available. And so happy to continue this collaboration into the future. Thank you very much.
Thank you, Alex. Great presentation. If anyone has any questions, please add them to the Q&A feature. We do have one from Cheryl, who also says, thank you for these updates. What a great resource for the community. But she's curious if the portal also includes future projections from the ROMspec model.
It does not.
Alex, do you want me to answer?
Yeah, please. Yeah.
So those projections aren't yet available. They're being performed right now, and but they will be made available, right? So they're not on the portal yet, but we'd love to see them up there and available to the community.
Thank you. Any other questions for Alex and her team? All good. Thank you, Alex. I will just note that Cheryl did go in and answer some of those remaining questions in the Q&A feature. So if you were interested on those, you can read up on those. And I don't know, Cheryl, if you wanted to comment on any of that. But we are moving into just a more broad open Q&A, and we'd like if our speakers could just turn their cameras on. And I think Kyla might have more of a broad question for you all, but also open if there are any other Q&A at this time.
Yeah, please feel free to raise your hand if you have additional questions, and we can get to those, or if there's any additional questions in the chat. This can be either specific questions for a specific project or a broad question for all researchers. I'll get us started with just like one general question for all researchers while other people might be formulating their questions, but we just want to know, based on what you learned and from your research projects, what would be, and you touched on this a little bit, but what would be a potential next step to further this knowledge in the context of policy and management?
I guess I can start. I mentioned this in the talk already, but we are finding that the fishery opening date does kind of fall right within when many of the spring spawning fish are still pregnant. So thinking about climate adaptive fishery closures where we're looking at current oceanographic conditions or actually like doing in situ sampling to see what stage the fish are in so that we can better protect those fish that are still pregnant, especially the bigger, older ones that we know contribute have outsized proportion contribution to the populations. So and then I guess I would also add another really great tool I think that we've started to produce is this environmentally informed stock assessment model for gopher rockfish that could potentially be applied to other species as well. And using that as a tool to better predict recruitment variability across years and also to help with management of the fisheries themselves.
And I guess building off that, other additional studies that you would perform to.
So we have started, one of the graduate students has extended beyond what I presented today to look at if we include environmentally informed models for other species. And I'm going to have a hard time like summarizing her results, but basically we are seeing really interesting patterns for some of the other species, particularly other spring spawning species. And so again, like if we can better predict recruitment, I think that is going to help with management overall across multiple rockfish species and not just gophers, which are less important to the fishery overall, more important to the recreational fishery. And John is raising his hand, so let's let him chime in too.
Thanks. Yeah, I couldn't resist. Because one of the things we'd hope to do with this project is, in addition to all the great work that was done, was to have a size component and to try and evaluate whether or not the larger, older fish had like a disproportionately more like buffer against these impacts or more of an impact, because we can kind of see it going either way. And the link to management there, I think, was going to be really interesting because that would allow us to look at the MPAs that the state has put into place for nearshore species, especially in the extra important role they might play, or the like less effective role they might play if, by contrast, the larger, older fish were more vulnerable to OA impacts in the context of how things are managed. And I know that right now, the Pacific Council is going through the process of removing many of the nearshore species from the federal FMP and delegating management to the states. Not my idea, first of all, but for better or for worse, it seems to be happening. And so in that context, kind of how the state manages those nearshore resources, especially the spring spawners that seem to be a little more vulnerable, is going to be really interesting. So, and long story short, there's a lot more that can and should be done to better understand both the impacts and how management might respond to this.
Awesome. Thank you for chiming in with that. It's interesting information and context. Do any other researchers have any thoughts on expansion of additional projects or research that could be built off these results. I understand that some are still finishing up, so it's okay if, we're not there yet with this question.
Well, I'll jump in and talk about the observational side of things. Is I think one, continuing it because time series grow in their power just by their length and by the number of instances of the annual cycle that we realize we'll see new things emerge. But this is that kind of mid-shelf zone is a place that's not typically well observed in terms of surveys, and yet has and the observing assets that we have up and down the coast. So I think that might be an important thing to be thinking about, expanding and augmenting some of the ocean observing systems with moorings of some sort. But where I think it's going to be interesting is seeing how we can, whether and how we can report that first big event. When we get that hypoxic event that actually looks like it could have a fisheries impact. And that kind of thing, because the real-time information I think can be pretty informative, as well as providing us. I'm looking forward to seeing how we can integrate it in, obviously, model predictions for what's going to hit the coast. See if we can get some forecasting there, supporting the validation of models when they finally do extend this far north. We'd love to see that ROMSPEC come around Cape Mendocino someday, and there was another one. Oh, kind of the idea of when, thinking about what drives HABs and things like that, having the ability to look into the water column at high resolution, when we're thinking about stratification and OA, PCO2, pH being things that drive phytoplankton blooms, but also phytoplankton stress and toxigenesis. Being able to do that and possibly couple it with some adaptive sampling or couple it with co-located IFCB or something like that to really get after those kind of questions.
I'm just going to weigh in quickly because we're running out of time, and then I'm going to pass it over to you, Christina, because I think it sort of relates. And not to caution, I'm always happy to hear about observations and more observations, but investment in additional data, even including experimental and research data, it needs a balanced approach with thoughtful consideration of data management and delivery. And I know folks at OPC and Sea Grant are aware of this, but I just want to flag that both are important, right? And so as these activities continue, making sure they're accessible to anyone who wants them in useful ways. And so down to Christina's work and a follow-on on the portal, I'd like to see potentially some better connection with the water resource control groups, the water boards, and thinking about discharge from POTWs and needs around water quality management and the nearshore, and how we could perhaps better utilize the portal and doing some analysis of observations, real time, real monitoring work, and some of the models, and perhaps also related to the HABs topic and HABs control interest. And so, Christina, I'll pass it down to you at Squirt for any last comments.
Thanks, Alex. I think what I would add is that these were really nice demonstrations, especially Cheryl on rockfish. We did a single study on Dungeness crab, but then we pieced together the rest of the framework and information from global studies of marine taxa from all over. And so really an emphasis on California's key marine taxa and more knowledge about what's important to California's, which fisheries and which species. Dungeness crab is, of course, very important, but then there's also the lower trophic food web and krill, and so much more information about the balancing experimental among species that are particularly important in the California current system, as well as nearshore habitats.
Amazing. Well, with that, we're at time. I want to give a big thank you and another round of applause to all of our speakers for sharing their wonderful work. And thanks everyone else for attending. So, yeah, thank you and hope everyone has a good rest of their day.
I thank you.
Thank you. Bye-bye.